Thank you very much. It's a great pleasure to introduce our company in COMEUP 2020 conference. Today, I'd like to introduce our technology as well as Seegene experience against the COVID-19. Our technology is very much optimized for the development of syndromic assay. Syndromic assay is the single test for all pathogens suspected of causing symptoms. For instance, if the patients are some symptoms of a respiratory infection, as you can see, there are so many different kinds of the virus and bacteria. Among them, if the patient taking syndromic assay, in this case, the three different pathogens are infected, including COVID-19 or influenza and then pneumonia. But in this case, pneumonia is the main cause of the symptoms. That's why this patient needs very urgent treatment for pneumonia instead of the COVID-19. COVID-19 indicating almost no symptom. That's why the molecular diagnostics providing very accurate information for proper treatment. So we are doing some campaign for the post-COVID-19. I will show you some of our campaign.
Students should be in school. Friends should rub shoulders. Neighbors should say hello.
나 다시 들어봐.
밥 잘 챙겨 먹고.
And families must not be separated. We're fighting a common enemy, and nobody knows its origin. But Seegene's molecular diagnosis lets us identify exactly where the enemy is hiding. This technology amplifies DNA by over 1 billion times and identifies viruses no matter how few in number. This winter, multiple viruses may attack at once, some even with unexpected mutations. But if molecular diagnosis becomes the new norm in our daily lives, viruses will never defeat humans. As long as we keep cheering for each other and fill our streets with songs of joy, humans will always prevail. Technology that protects our everyday. Seegene.
Yeah, as you can see, until today, most of people did not know about the molecular diagnostics. If you are taking molecular diagnosis in your daily life, I believe it will change your life. First, I will introduce what is our technology. So Seegene developed the world's first AI system for the development of the multiplex molecular diagnostics assay systems. So we have 3 different technology groups. First one is a multiplex technology for syndromic assay. Another one is a development technology for automatic assay development. Last one is how we can utilize our technology with all over the world scientists that we call bioplatform through the internet. You may be familiar with the real-time PCR. So previously, you don't know what is a real-time PCR. Now, most of the people have a chance to know what is a real-time PCR. Real-time PCR is used for COVID-19 test. So I mean the real-time PCR is a golden standard method in real-time the molecular diagnostics. Most of the other molecular diagnosis companies, they are using TaqMan method. TaqMan method was introduced in 1990. It means the technology was developed more than 30 years ago. So our technology recently developed. So only our technology can generate the syndromic assay. TaqMan technology, single assay, maximum 4 different target or 4 different pathogen can cover it. Our technology can be up to 25 different pathogen or maximum 15 different pathogen or 25 different pathogen in the single reaction. So we are using the 5 different representative technology. One of the example is applied to the cervical cancer diagnostics. So HPV, human papilloma virus, is main cause of the cervical cancer. That's why if you can check it up about the presence of the cervical cancer or not, you can suspecting the woman can have an opportunity to taking cervical cancer. That's why the lady has to taking HPV DNA test during their life. And then they have chance to avoid cervical cancers. Here is introduced some the global companies product, Hologic or Roche and BD. They use the real-time PCR-based HPV DNA test. Only Seegene providing full information, but other companies are providing partial information. We call targeted assay. We are syndromic assay. That's a huge different information we are providing compared to the other technology, our technology. Now, I will introduce about our automatic systems for assay development. Until 2015, we used the chemistry technology or DPO, another TOS or MUDT. So DPO is I'm inventor of this technology. So at this time, only 1 technology, 2 technologies, 3 technologies for assay development. From now, combined with the BT, NT is combined together, we generated some automatic assay development systems. In this assay development by AI, integrate with 25 different proprietary technologies. As I said previously, our R&D people manually assay development. Now, digitalized assay development. It means, in the camera, long time ago, they used a film. Now, we are digital camera. So assay development also, we are using digital approach, mean the huge number of the assay development by digital approach. As you can see, it's a very complicated process. Also need some technology, experience, and know-how. So this is current our environment. All of the molecular diagnostics company doing this kind of approach. So our technology, experience, we transfer to the computer. So 895 parameters. It means a kind of the decision point. So the R&D people has to know about this number of the decision point. How people can memorize conducting assay development is quite difficult. That's why depending on the R&D people, quality is slightly different. So if a computer can take over, best quality of assay development that they can conduct. So we call AI-based assay development. And then the AI providing all necessary tools for the assay development informations, such as the oligo design or optimization or verification. Only R&D people can do sample collections or the optimization or product verifications. And then product complete. So this is a normal process, step 1 to step 16. This is the long period. Although in here, just the 16, each step, sometimes 3 years experience R&D has to conducting. No experienced R&D, they cannot do that. So normally, one assay, syndromic assay development is taking over years. So AI-based or automatic systems, without the special knowledge or some skill or know-how, they can eliminate all kind of the complicated process. This is AI-based the assay development. Its good thing is computer, AI, does not complain, even does not asking vocation. But the human being, they always, sometimes something wrong, if somebody left, we have to replace the training. So computer and AI, just they are doing whatever we need. This is one of the demonstration. The molecular diagnosis laboratory required so many different kind of instrument for many different assay systems. For instance, here, 16 different instruments for 77 different assays. Our approach is replacing just one platform, cover all kind of the syndromic assay. For instance, in the home, if you have a television, one television, one channel. So if you need 10 channel, you may need 10 different television. How about 100 channels? You may need 100 different televisions. So this is a long time, a kind of the analog the approach. How about the digitalized the assay development? Just the one instrument systems can cover all kind of assay. That's the future directions. We are doing this kind of approach. Another one is how we can utilize our technology and then automatic systems through the internet. Until today, not only Seegene, every company in the world, molecular diagnosis company, they are making assay by themselves. It means that their own R&D peoples. So our approach is different way. Although we have a very good technology, how we can maximize by our R&D people by working with the other scientists. So other scientists mean in the other hospital or university, so many scientists, expert is theirs. They can using our technology for assay development. We are providing our AI tools for assay development. And then, but also we can providing grant or raw materials. And then the customers, mean the scientists, the expert, can develop the assay by supporting by our guidance. So develop the product that they can providing us for commercialization. And then we can sharing our profit, just like a royalty. This is our approach. You can imagine if this technology are using our company only alone or sharing with the worldwide scientists, how many products we can generate. We cannot be expert, not only human or plant or animal or the ocean or food science. So if we are sharing our technology with all over the field, so 100,000 assay system we can generate. Current existing platform and Seegene platform is a big difference. Most of people say the internet-based platform is it's the same kind of the platform. No, that's not the case. Facebook is Amazon and Google and so on are consumer-based network. How about the Seegene platform is some assay development-based network. It means only expert can be involved. So specialized product development. The other hand, only specialized company can handle this kind of platform. The Seegene is some assay development platform we are providing, we are sharing with the worldwide scientists. We prepared about the 60-second animation for understanding about your approach, AI-based assay development. As you can see, our platform covering all kind of the area, not only medical side, but food or quarantine or the ocean or the plant. All of the field scientists are working together. Now, I will introduce the Seegene experience against the COVID-19. This is my note from the January to very early this year. We started the assay development January 13, and then within 2 weeks, we finished the assay development. Another 1 week is taking some documentation or experimental data and something in order to submit our assay developments to the Korean FDA or CDC. In this case, we submitted the assay system to Korean CDC to get the emergency approvals. We got the February 12, we got the emergency approval. This is taking only 1 month. That's compared to some other country, very short period of time, we finish up the assay development and got the approvals. The first, our approach is most of the other companies are making a single assay. That's why 2 different targets, they need 2 tubes, but we have 3 different targets in single tube. That's why our approach is very efficient, the detection of the COVID-19 compared to the individual. We made some contribution to the control of the COVID-19 quarantine in Korea. As you can see in this figure, the blue color is indicating the accumulated test number. Red color is a positive number. So the compared to the blue and the red, positive ratio is about 1%. This is very low compared to some other country. How we are making very aggressive tests in the single laboratory. This is a workflow for the COVID-19. First step in the field, there are sample collection, and then arriving to the test laboratory. Step 2 to 6, this is the only laboratory they are conducting. First step is RNA extraction, and then another PCR reaction or the interpretation, and then reporting. Compared to the other country, to the Korea, we have a very big advantage. First one is an automatic system, step 2 and 6. Another one is a centralized medical systems. Centralized medical system means in one hospital taking all kind of the assay in the single laboratory. That's why automatic systems, centralized laboratory systems combined together allowed us mass testing capacity. This is a huge advantage compared to the other countries. So most of countries, they did not do the really attention about the speed of testing should be faster than the speed of transmission. This is extremely, extremely important for the control of the pandemic, especially the COVID-19. Why this is so important? Some of country result is coming from 1 week behind, or sometimes 10 days behind. This is too late. In Korea, most of the case result is coming within 1 day or occasionally 2 days, but most of the case within 1 day. This is very important. Why rest of the country is struggling because of they did not really control about the speed of test. As I said, the different thing is between Korea and other country. We are centralized lab medical system. Other is a decentralized medical systems. Centralized medical system has advantage, aggressive investment for the some instrument or some quick introduction of the advanced technology or unification, especially for mass test capacities, need a centralized laboratory. But some other countries, one hospital, each department, they have their own test laboratory. In the May, we have another product launched on the market. because the COVID-19 has a mutation. That's why the people are afraid our current test is okay or not. That's why we added one more target gene, total 4 different target genes. The increasing coverage of the COVID-19. Another advantage, extraction process eliminated. But at that time, some RNA extraction reagent shortage, this was a worldwide big issue. So if we can eliminate this step, we can saving cost or time, also effective solution for reagent shortage. Very recently, we also introduced another COVID-19, including influenza. So the people are afraid about the twindemic issue, not only COVID-19, but also the influenza. Most of the approaches of COVID-19 or influenza, also we covered the common cold. RSV is a very big issue about the common cold. Another advantage is checking up the condition of the sample collections. This slide is show the detection pattern of the seasonal flu and also common cold, RSV. So as you can see in here, the blue color or pink color is influenza or RSV. Is annually pattern is keep coming again and again and again. COVID-19 also similar pattern. That's why most of the country are concerned about the twindemic or triple-demics. So, but between the influenza, RSV, or the COVID-19, symptom is very much similar. Without the molecular diagnostics, doctors cannot distinguish. Compared to the companies, the assay system, only single COVID-19 or influenza A and B, but our approach is 2 additional COVID-19 and also the RSV, another the internal control for checking up the sample collection. This is compared to the current approach, our approach is big different. Our approach is only Seegene can make this kind of product. Some existing TechMan technology does not make these assay systems. Now I have a last animation. Before I closing up, before I finish my presentation, I like to emphasize about the importance of the molecular diagnostics. From now, you will have chance to take in molecular diagnostics. So as long as you are taking molecular diagnostics in your daily lives, you can make a very healthy and happy life. Enjoy yourself. It's taking about 30 or 60 seconds. This is my presentation is done. Before I complete, whenever you have some symptoms with some infections, so from now you can visit in the nearly local clinics and then identify what's cause of symptoms, and then you can take a proper treatment. So I believe molecular diagnostics make you very healthy and happy life. Thank you very much.